Good evening. I'm Linda Schott. I'm the president here at the University of Maine at Presque Isle, and I'm thrilled to see all of you here tonight. This is another of our series, the Distinguished Lecture Series. We have brought such wonderful speakers to the campus and the community over the last 15 years. In fact, this month is the 15th anniversary of the Distinguished Lecture Series. Could you join me in a round of applause to celebrate? I know since I've been here, we've had a variety of speakers. Before I came, there were astronauts as well as journalists and other kinds of scholars. So I think this is really one of the marquee events that the University of Maine at Presque Isle does, both for our students, faculty, and staff, and for members of the larger community. Several members of our campus community work very hard to bring this to you, and I'd like to call their names in case they're here. If they're here, would they please stand and be recognized? I'm pretty sure that the leader of the group, Dr. Ray Rice, who is our interim provost and vice president for academic affairs, is not here. He had to go down to Machias today, and he's been furiously driving back, but he hasn't made it back yet. Committee members include John DeFelice from history, Tomas Herzog from education, Vanessa Patnod from physical therapy, Chenzeng Wang, I know Chenzeng's here, from Mary Lawrence, who works here in the campus center, Vanessa Pearson, who works with student activities, Gayla Shaw, who works in Rachel's office, and Rachel Rice, our director of public relations. It takes these folks a lot of extra time to brainstorm the ideas, think about who exactly are the right speakers for the series, and then to make all the arrangements that go into getting someone here. So thank you to the committee for all of your work doing that. Tonight, we're going to have a chance to hear about some of the latest cutting-edge work being done here in the state of Maine around cancer research. As a cancer survivor myself, this is, as you would imagine, of great interest to me. I'm looking forward to hearing more about what Carol has to say so that I can think about how it might have made the decisions that I made last year different, how it may make them different for some in the future. I would also note that this is about individualized cancer therapy, or I've also heard it called personalized or personalized medicine. And we here at the university are, in our new vision, talk about personalized education. It is the wave of the future. You know, we all expect, whether we're on our iTunes account or on Amazon, we expect those personalized recommendations that can come with greater and greater technology and sort of maybe sometimes a little bit scary monitoring of everything we're doing out there on the internet. So I think there's an interesting tie there between our vision of personalized education and working more and more in terms of each student's learning interest and learning styles, a program that will really kick into gear next fall, and what's happening in medicine as they move more and more to thinking in terms of individualized treatments and therapies. But that's about as much as I know about that, so I'm going to turn it over to somebody who knows a lot more. Please welcome Dr. Judy Rowe from our biology faculty to provide more details and do the official introduction. Judy?
Okay, I'm also thrilled to see everyone here. I'm Judy Rowe, assistant professor of biology here at UMPI, and like everyone else in the room, I'm sure you have a connection to cancer. Everybody does, don't we? And personally, I am the daughter of a pathologist, so he's very aware of all of the cancer around. So, you know, I'm sure this talk is going to be of interest to everyone. And we're really honored tonight to have our guest distinguished lecturer, Carol Bult, and she's here from Jackson Labs, which is in Maine. And Dr. Bult has actually been there for 16 years, where she's recently become the director of this new Cancer Avatar Individualized Cancer Therapy Research Program that she's going to talk about. And one of the reasons I wanted to invite her is because she's a scientist here in Maine, and I was hoping to be an inspiration to all of our science students and all of our students, because it's one of the, it's an internationally recognized scientific research institute. It has top scientists in the world working there, and actually, Jackson Lab was actually started by a University of Maine president, C.C. Little, in 1929. And it has now grown into one of the most important research centers that uses mice as a model for human disease and human health. So the other thing that the lab does that maybe people aren't totally aware of, as scientists we are, but the lab has also been a major force in generating what are called genetic resources, which are available then for other scientists around the world to be doing even more experiments. So Carol started her career as a plant population geneticist, actually, and she worked for a time at the Smithsonian Institute in Washington, D.C. And then she was hired by what was called the Institute for Genomic Research, or also known as TIGER. And this was a fledgling operation, I'm calling it, headed by a man named Craig Ventnor, a scientist. And if you know his name at all, he went on to become the second project on the human genome and was able to do it much faster than the project that was ongoing. So he was a pioneer, and Carol was there at the start of this. She was involved in the first, was it three organisms that were sequenced, completely sequenced. So she was right there at the time when it was all starting. And then, so we now have gone through a revolution. So there's a new genetic revolution, genome sequences, there's huge quantities of DNA, sequence information, and this has really required completely new approaches to information processing, databases, analysis, and Carol has been at the forefront of using these approaches and developing these approaches down at Jackson Lab. And in particular down there, she spearheaded what is now known as the Mouse Genome Informatics, MGI. It's a lot of letters and everything we do. And so in that case, it was to, well, you have the mouse DNA data, what are you going to do with it? And her group has been at the forefront of figuring out how to use this information, and they've even said, you know, for the students, that information processing and et cetera is going to be the new job in the future. So I hope you get some inspiration from her today. And as was mentioned, tonight she's going to tell you about their latest efforts to understand human cancers and to improve the understanding of the complexity of patient responses to the therapy using what's called genome-based information and specialized mouse genetic systems. So welcome and thank you for coming, Carol.
Thank you for coming out and spending the evening. It's really quite an honor to be part of this distinguished speaker event and to have a chance to come up and visit. This is my first time in Presque Isle. I usually exit at Medway to go to Baxter. So now I know where the north entrance is. And I really look forward to coming back when it's just a bit warmer. So. Yeah, so tonight I'm going to, as it's been mentioned, I'm going to tell you what we're doing at the Jackson Lab in the area of taking this basic science that we're known for and trying to translate it into a more clinical realm. But I know a number of you may not know as much about the Jackson Lab, so I wanted to say just a few words about it. So we are a not-for-profit biomedical research organization. I think Judy mentioned we're founded in 1929. We actually have 3 campuses. We have the Mammalian Genetics campus in Bar Harbor. We have a facility actually in Sacramento, California that uses the mouse resources as outreach to pharmaceutical and biotechnology companies, largely for drug discovery aspects. And we have a brand new campus going up in Farmington, Connecticut, that's going to be focused on human genetics and quantitative cell biology. So we are really a rapidly growing enterprise. We've been around a long time, and we're in this growth phase for our institution. We currently have 45 principal investigator-led research groups, and the areas of research are actually quite diverse: neuroscience, computational biology, immunology, and cancer. We were founded as a cancer research organization in 1929. We also have a lot of outreach. So in addition to research, the other main mission of the lab is outreach and education. We have an extensive courses and conferences offering. We have internships, pre- and postdoctoral training, visiting scientist, investigator program, a lot going on in the area of outreach. And then finally, we are a resource. So all of the information, a lot of the mouse resources, these are distributed to biomedical research facilities around the world. We have about 4,000 different types of genetically modified mice. It's the most diverse vivarium in the world, and we send those mice packing to over 16,000 laboratories all over the world. So this small little research facility on the coast of Maine sees the world. So, but the most important thing is our mission. And the mission of the Jackson Lab has always been to try to really understand the genetic basis of human biology and disease using mouse as a model system. So I'm going to talk mostly tonight about this in the context of cancer, even though we use mice to understand many of these other biological and disease processes as well. And it turns out that mice are really quite excellent model systems for understanding human biology. And in cancer, they've been used to as a test bed for clinical development of new drugs for cancer therapies, for understanding the basic properties of the most problematic aspects of cancer, including metastasis. They've been used to understand drug resistance and to model early detection of cancer. They really have contributed in major ways to all of the current practices for cancer treatment, and as I'm going to show you, are going to continue to have that influence, I think, even more. So one of the things I'm often asked is, well, mice are models, but mice aren't human. Right? So mice really aren't human. There are things about mice that don't match what goes on in human, and there are things in human that don't match what goes on in mice. So I want to emphasize that mice aren't good models for everything, right? This is one of the examples where mice don't model things. This is actually a real magazine, by the way. I don't have a subscription, but there's a lot of people out there that try to make mice human in very strange ways, and this is one of them. But the point about models is that model is a lie that helps you see the truth. And this was a quote from Howard Skipper, who was a biochemist and a cancer researcher in Alabama. And this is really one of the fundamental truths. This is why we can use C. elegans, which is a nematode. We can use zebrafish. We can use yeast to get insights into basic biological processes that have relevance to human biology and disease. And we understand that these are models, but they are models that help reveal the truth of biological processes. So tonight I'm going to tell you a little bit about how we use the mice in what we're calling a patient-derived xenograft and avatar program. And the goal of this program is pretty audacious. It is to come up with ways to predict the best drugs to use for an individual patient's cancer. It's really trying to understand some of the really thorny problems in cancer biology, including drug resistance, which is one of the key problems in cancer treatment, is that when if the cancer comes back because it develops resistance to the treatment, what do you do next? So how does cancer cells, how do they acquire that resistance, and how can we overcome that resistance? And this whole idea of these personalized cancer diagnostics. So all of this applies sort of clinically relevant questions. These at the Jackson Lab are all based on a really solid foundation of basic biological research into cancer cell robustness, so how cancer cells use cell machinery to ensure their own survival at the expense of other cells. The whole genetic and genomic complexity of cancer, and progenitor cell biology or stem cell biology. This is also a very important aspect in understanding basic cancer biology. So on that platform, we're building this program that will enable us to use the mice as this bridge, translational bridge to individualized cancer medicine. And that's what I want to talk to you about tonight. So the basic idea about what we do is we use a specialized mouse called the NSG mouse. I'm going to tell you a little bit more about that mouse in a minute. And the idea is that we transplant human tumor material into those mice, test drugs in those mice, and then use that to guide patient care. That's kind of the whole big picture, and I'm going to show you some examples of how we do that. But let's start, before we go there, to the science, let's start with the current cancer treatment paradigm. So this is the image that I like to use. So oncologists, and we have some in the room, so they can speak to this much better than I can. Oncologists have often numerous treatment options available to them for their patient. And they pick what they think is going to be the best one, and sometimes it hits the bullseye. It does the job. Sometimes it doesn't. And so you got to take another arrow from that quiver and try again. And sometimes it works, sometimes it doesn't. And it's great that there's all these options, but what if we could pick the option that we knew was going to have the best likelihood of hitting that target the first time? And it didn't become such a trial and error process. That's one of the things we want. We want targeted, we want tailored therapies. The other aspect about cancer biology that is particularly problematic is this whole issue of relapse and resistance. So oftentimes the treatment will work for a time, but that cancer will come back. So can we be prepared during that interim time of good health for the patient? Can we start to get prepared for what's going to happen to that tumor in the future and pre-select treatments that we think are going to be the most effective for that patient? Is this all possible? So we do this, like I said, through taking human tumors and implanting them into these mice, and this process is called xenografting, xeno being foreign, right? So we're taking human tissue and we're putting it into a different species, we're putting it into mouse, so it's a xenograft. And one of the reasons that we're taking this particular tack in cancer research is that a lot of work has been done in cell lines, so growing cancer cells in plastic petri dishes. And that's a very unnatural state for cells, and the responses you get to drugs in that particular system don't always mirror what you see eventually in the patient, because it's such a foreign, it's a monolayer of cells. Well, cells don't exist as monolayers, right? The tumors, they just don't do that. So by doing these xenografts, we think that we're mirroring the cancer biology much more faithfully, so that the observations we have on the human tumor in this system is going to be more biologically true to the original tumor from the patient. We can also use this from a pharmacology perspective. The response that we see in these xenografts, again, we think are going to be much truer to what you're going to see in a patient than you would ever get from some other system, a cell line system, for example. So this actually was a paper that was published by a collaborator of ours, let's see, about 2 years ago now. And it's kind of a complex figure, but really what you have to look at is on the far end there, I have a pointer. On the far end, here's your patient. These are all the different things that characterize the patient with refractory cancer, cancer that's resistant to treatment. And these are the tools that we have to address that cancer situation. And cell lines, which are very common, they really don't do a very good job at modeling human cancer. They've been used for quite a while because they're relatively easy to scale it, easy to do it, but the biology is just not faithful to what you see in the patient. So we can actually put cell lines into mice, so cell line xenografts, And that's a little bit better. It moves us a little bit closer to the actual patient condition. But there are still characteristics here that actually don't model what we see in the patients as directly as we would like. And so the patient-derived xenografts, which I'm going to tell you about tonight, it's a mixed bag, right? So they don't model everything about the human condition perfectly, but they get us a lot closer to being able to model the human disease in a much more realistic, translatable way than you can with any of these other sorts of approaches. Among the best ones are that the tumor heterogeneity, so tumors are complex mixtures, that is preserved in these xenografts, and so that's really important for trying to model what's going to happen in a patient. But what's still sort of limiting us and what we're working on is that the immune systems in these mice are not human immune systems. And so it's really one of the things that we have to work on over time is to move this bar closer to the human system. And I'll talk a little bit about some of the work we're doing to make that happen. So that's all background. These are the mice. These are the NSG mice. So this is really the foundation for this whole concept of cancer avatars and personalized medicine. Without these mice, we wouldn't be anywhere near where we are in reaching that vision of individualized medicine for cancer. So these mice have been genetically altered to have no immune system. They have none of the natural components of the immune system. So what that means is that if you put a human tissue into this mouse, it's not going to reject it. It has none of the cell components that needed to mount that response. And we can use that to our advantage because they become a host, a transplant-compliant host for human cells and tissues. This is not a new idea. So xenografts have been around for a long time, and in fact they have for a while, a really bad reputation, because they weren't that great models. They've been around since the 1960s. But what's happened over these many years is that scientists have been working to make additional modifications, genetic modifications, to get these systems more and more compliant to being able to accept human cells and tissues. And this is our current and best model, these NSG or non-skid gamma mice. And as you can see, it's taken a long time to get there. And this mouse was developed by one of my colleagues at the Jackson Lab, Lenny Schultz. And he actually didn't develop this for cancer at all. He developed this to study the immune system's role in diabetes research. So it came out from left field. This mouse is now the mouse that's used in cancer research of the type that I'm talking about today. So what's the benefit about this? So we want to transplant human cells into these mice. These are the older models, and you can just tell by these indications of how well different cells and tissues take in these different immune-compromised mice, that NSG certainly far outperforms any of these other current forms of immune-compromised mice out there. So it's really the current and best that we have, but it's not the end of the story. I think in another 5 to 10 years, we'll have a number of different additional modifications to this genetic background that'll make it even better. So our whole PDX and Avatar program has basically 4 components, and I'm going to tell you about those 4 components tonight. The first is a component where we're making a publicly available PDX resource. The second is where we're taking this approach of xenografts and using them to create a model from an individual patient for the purpose of doing the testing in the mice to inform what's going to work best in the patient. And then I'm going to tell you about the Avatar, which is really integrating a lot of different components of all of these things into a program that we think is going to be the most clinically relevant in the future. And then I'll talk briefly about some of the innovations that we're working on to make this system really much more realistic, much more cost-effective and time-effective, given what we know about the natural history of cancer progression. So let's start with the first one, this whole PDX resource that we're developing. So let's start from the beginning. How do we make these PDX models? What does that mean? The process is actually pretty simple. A surgeon isolates tumor material from a patient, and they send it to us in a buffer, and that tumor gets implanted subcutaneously in the right flank of one of these host-compliant mice. These NSG mice don't have a functional immune system. That tumor then, because it's all together with all its friends, continues to grow, right? The cells continue to divide, and that tumor is actually going to grow in size. And when it reaches a target volume, we take it out of the mouse, we characterize its genome, we cryobank some fragments, and then we take some and we put them into between 10 and 20 new mice. So we go from a passage 0 to a passage 1. So now from this one patient's tumor, you're now going to have up to 20 mice from one of these P0 mice. You're going to have now 20 to 100 mice that are all carrying, all tumor-bearing mice, are all carrying tumor that's identical to this patient's tumor. So I'm building up this resource of all these tumor-bearing mice. And then when those mice, when their tumors reach a certain size, I'm going to bank it. I'm going to put it in store it in a special way, in a repository, until I'm ready to do a study. And when I'm ready to do a study, I'm going to take those tumor fragments out of the freezer. I'm going to put them into more mice until I have enough mice that I can do a statistically robust drug study. So now I'm doing a drug study on hundreds and hundreds of mice. I can test many different kinds of drugs. And I'm going to see if that's going to mirror what's going to happen to this patient. And I've got a couple of examples to show you later on. But in order to do that kind of work, I have to build this resource. I have to build this bank of many different kinds of tumors so that when we want to do a study, we can just look up in the database, well, I need all the lung tumor models that have a particular mutation, and I can pull those out of the freezer and grow them up. So what do we have to date? We have about a little over 300 models. So those are mice in which we put tumors and that successfully passage to that P1, and I've got them in a bank. So those are 300 established models. I've got almost 300 in progress right now. But look at this number. I've got 777 that we tried to implant that didn't take. They didn't grow. They failed to grow in that first passage. And for the types of cancers that we have, we've got about 20 different types of tumors. Most of the models in our bank are from lung, brain, bladder, breast, colon. It's quite a diversity, but we're trying to focus now on a few of these and really build a really definitive bank of tumors for that type of cancer. And we don't do this alone. This is done in partnership with a lot of clinical centers around the country, including right now in Maine. Actually, Eastern Maine Medical Center is number 2 in our repository. The only cancer center that sent us more tumor materials to build this bank is the University of California, Davis, which is a comprehensive cancer center. They see thousands and thousands and thousands of patients. So Maine is well represented in this resource. So Eastern Maine and Maine Med are both Maine medical centers that are contributing to the development of this resource. So it doesn't always work. Okay, so how often does it work? About 50% or so, 55% don't engraft. All right, that seems pretty high, right? But this is for all cancer types that we try. So of all the different cancer types we've ever tried, when we put them into a mouse, about half the time it's not going to work. It's not going to take. It's not going to grow to that target volume. So this is what didn't work, this is what worked, and this is what is in progress and is likely to work. So we want this to get bigger and that to get smaller. But this is for all the tumor types. If we look at some, like colon cancers, we've got a really good success rate. almost 50%, maybe a little more than 50% of these tumors take really well in the system. So if we engraft them into the mice, they grow really well, which is great. And then we've got the counterexample, which is prostate cancer, in which, number one, it's really difficult to get the material, and number two, even when we get the material, it doesn't work. We can't get prostate tumors to grow in these mice, right? And I'll tell you a little bit what we're trying to do to solve the problem a little bit later on. So currently, not every cancer works equally well in the system. The ones that work really well are the larger tumors, the ones that are more advanced tumors, the higher grade metastatic lesions take really well in the system. But the NSG has helped us because compared to all the other immune-compromised mice types out there, there are certain tumors that will grow in the NSG mice that won't grow in other types of systems. Some myelomas, certain types of breast tumors, melanoma, AML, we have a big program in AML, acute myeloid leukemia. So it's a great system, it's not a perfect system, and so part of our innovation is to improve how the system works overall. So another question I get commonly is, so when you put this human tumor in a mouse, does it change the tumor? Is it really still a human tumor, or does putting it in the mouse change its biology significantly? So we have a pathologist, we have input from a lot of pathologists. We can't do this work without surgeons and oncologists and pathologists. So we have a pathologist at the University of California who actually looks at this question for us. This is a lung tumor model that I'm working with right now, and this is from the patient. And this is from the tumor from this patient that's been engrafted into one of these NSG mice. And from the morphology level, many of the features that you see in the patient tumor are recapitulated in this engrafted tumor. And the pathologist tells us that this is a really high concordance, right? So that all looks good. It doesn't always work. This is another one of the tumors I'm interested in. And here's the patient tumor. When you put that in the mouse, it's not a really good concordance. It really looks like something has changed here. Maybe the part of the tumor that got engrafted in the mouse is different than the part that got sampled for this pathology image, but the concordance is not always perfect. We also worry that when we do this engraftment, that we need to make sure that the system, that tumor that's growing is actually human. So we have a number of different ways of doing that. We do this, different kinds of stains under the microscope can tell us whether or not the tissue we're looking at is human or not. So we do this standardly for all our tumors. So if you see this kind of brown staining here, these are actively dividing cells. And this particular antibody only stains human cells, it won't stain mouse cells. So this is the patient tumor. These are the actively dividing cells, and here's the engrafted tumor. So again, we've got human cells, and they are clearly actively dividing. So we have some checks and balances to make sure that what we're actually working with is in fact human, and that it looks like the patient tumor did. Important stuff for looking at the biology later. So I wanted to tell you, I don't have time to tell you all the really cool things that are happening, but one of the things I mentioned was I showed you that really depressing graph about the poor take rate of prostate cancers. So it turns out that this is one of the places where mouse and human differ. Prostate cancer cells require a very specific molecule called a cytokine to grow. And the name of that cytokine is IL-6. And the mouse IL-6 does not work in the same way as the human IL-6. So we aren't going to get prostate cells to grow in mouse because they're not going to get the right signals to tell them to grow. So what can we do? Well, what Lenny's doing is he's taking a human IL-6 gene and he's making human IL-6 transgenic NSG mice. So he's going to take those immune deficient mice and he's going to make them express the human version of that gene. And by so doing, we're going to make it possible to use this system to do this kind of work in prostate and in myeloma. And there's additional types of tricks like that, genetic tricks that we can do to the mouse genome because it's so experimentally tractable, to keep improving the system so we can model more types of human cancers using this approach. We're also building NSG mice that actually have human immune system components in it. So this mouse doesn't have its own immune system, but immunology is a really important part of cancer treatment and cancer response. And so if we can actually get these mice to express human immune system cells, that opens up a whole new area of research where we can try to figure out how to get our own human immune systems to do a better job at fighting cancer cells. Again, really exciting stuff. And if you're interested in these other approaches, we can talk about them at the end. But there's a number of things we're trying to do to make this thing really work effectively for a broad range of cancer types. So we have this resource, and we're constantly innovating to make the whole system work better. Does it actually work? So now I want to tell you a little bit about this part of the program called PDX Patient, where we actually exercise the whole system. And the idea is this, is that we're going to get a tumor from a patient, and remember, we're going to put it into the mice, and we're going to passage it a couple times until we get hundreds of mice carrying that tumor. That was from previous. So once we have that, once we have a bunch of mice all carrying the tumor from a patient, then we can start testing it with different drugs and different drug combinations and watch what happens to that tumor. Does it stay the same? Does it shrink? Does it grow? Does it go away completely? And all of this we can do in the tumor-bearing mice without subjecting the patient to this sort of experimentation. So this is like a virtual clinical trial for an individual. And the idea would be, this seems to work the best, right? So maybe this is the drug treatment we should do on this patient. We shouldn't probably use this one at all. And that's the idea behind doing this. So this just sort of restates, summarizes that whole thing where we start with the tumor, we put it into some mice, we let it passage, we put it into more mice, create a big cohort, and then we can start drug testing. But notice the time it takes, right? So it can take, it takes on average about 6 months just to get this tumor to grow in the first passage. And then it takes another 2 months to create the next cohort of P1 mice. And so it's about 10 months from the time we get the tumor before we can start a drug testing. So that time frame really limits in many ways how we can apply this realistically right now. It's one of the areas we really have to innovate. This really has to come down in time. So does it work? So this is a case of some collaborators of mine from University of California, Davis, David Gandera and Phil Mack. This is from a patient, she was 44 at the time of diagnosis, never smoker, lung cancer. And when she came in to David's group, she came in here, so this line, this indicates her, it's kind of a graphical illustration of her tumor burden. So the higher the peak, the worse off she is, the more tumor she has. The lower means she's got a good response. So she got diagnosed, chemo didn't do a whole lot for her, but then they found that her tumor had a particular genetic mutation in it that made it susceptible to a targeted therapy. So she went on that targeted therapy called erlotinib, and you can see that she responded really, really well to erlotinib therapy until that resistance came back. And now her tumor burden goes back up. And then she had some metastatic disease. And about this point, the tumor burden is really quite high, and she went on this experimental therapy of afatinib and cetuximab. And she responded really, really well to this therapy for a while. She has since succumbed to her disease, but this experimental therapy, she was clearly a really good candidate for this. Now, from the PDX side, we got her tumor late, right? So we got her tumor after the resistance had been acquired. and we created the mice, the P0 to P1s, and when we got to about P2, we started the drug treatment, and really the question was, is what we're going to see in the mice going to mirror what happened for this patient? And so this is actually now the tumor growth curves for the mice. So this is the tumor-bearing mice, bearing her tumor, treated with the same kind of drugs that she got treated with. So here's the control. So this is just saline, and you can see that the tumor volume just goes up and up and up. That's what we expect because there's no treatment. This is her treatment with erlotinib, and remember that she was originally responsive to erlotinib, and then resistance came. And so when the tumor first got treated, it actually dipped a little bit, but then here's the resistance, no longer responsive to erlotinib. And then here's the treatment with the afatinib and cetuximab, and what we did is treated both singly and in combination. And this tumor would have been considered, this is a stable disease. This would be our indication of a stable disease. So actually what happened in the mouse, in this tumor-bearing mice, is exactly what happened to this patient. Had the exact same profile of response to the drug. So had we been able to get the tumor earlier and set up this trial earlier, we might have been able to test all these drug combinations and have known earlier what that patient could have responded to for her second-line therapy sooner. That's one of the goals that we had. So this is a really great response curve. This is really what you want to see. Do we always see this clear response? So I have another case. This is an ovarian patient who has a late-stage ovarian cancer, stage 3. And she has been through quite a bit of treatment for this disease. Many cycles of chemotherapy, sometimes with partial response, and then sometimes with progression, and then stable disease, and then metastasis. It's kind of that same curve that you saw previously. And so when we first got her tumor, which was in 2012, I believe, we implanted it into the mice, and at that time she was going on this Doxil treatment. And what we wanted to see is, would the tumor from her engrafted in the mice respond in the same way that she responded? So we had 3 treatment arms. We had an untreated, we should have had Doxil plus carboplatin, but for a number of reasons we just had Doxil. And then we had carboplatin plus paclitaxel, which what we predicted is that we should get stable disease with Doxil, but we suspected that either the tumors would not respond or that they would lose their ability to respond to this particular treatment. And that was kind of based on what had been observed for this particular patient. And this time, we were interested in the problem of tumor heterogeneity. So when we do this work, is the result we're going to get depend on which tumor we pick to progress on to the P1 animals. So we took a bunch of different P0 tumors and created P1 mice, and all to see how reliable the response is. So, and this is the results of that study. So we did 3 different tumors. There's actually P1 tumors, but they were from P0 tumor A, P0 tumor B, and P0 tumor C. So there's 3 different groups of tumors. They're all getting the same treatment. These are the vehicles, so as expected, the tumor is growing away. But these are the 2 treatment arms, and you'll notice that it's really actually very difficult. The response here is kind of ambiguous as to whether Doxil and carboplatin plus paclitaxel actually give you different responses. And you can't tell from the graphs alone, you have to do some statistics. And so when we do that, we did 2 studies. These are the same mice. We checked them at 28 days because that's what we standardly like to do. And it was looking like the treatments weren't actually, at 28 days it looked like stable disease for both treatments in 2 of the 3 tumors. Only one showed kind of what we had expected, which was that paclitaxel plus carboplatin that that tumor would progress and Doxil would be stable. So only 1 out of 3 tumors was giving us the results that we expected. So we let the study go out another couple of days, to another month or so, and out to day 59. And then 2 of the 3 tumors showed what we expected, with this treatment showing progression and this treatment showing that it was pretty stable. So now this is really going to be, I think, a very common thing where you don't get that blinding insight, where it's just a little bit ambiguous what the results are telling you. And one of the challenges that we have to work with the clinicians on is, how do you interpret this? Are you going to base a patient treatment on data that are less than clear-cut like this? And how often are we going to run into cases like this where it is not as clear-cut as the case I just showed you, where the difference between treatment and non-treatment was enormous? We don't know yet. This is part of why we're doing the science. And then if we can understand why some cases we see the big gap and some reasons we don't, I think that'll help us a lot. So we're doing a number of these studies for other cancer types. I'll tell you a little bit about the breast cancer work we're doing in a minute. But this is the kind of thing that we're facing. It's not always going to be clear-cut, but we fold the questions back into the science and then the science back into the translational research. So before I tell you a little bit about the AVATAR program, I wanted to say just a few words on the genomics, on the DNA, tumor DNA. So this is a screenshot. Ellen, you probably know what this is. This is from a resource at Vanderbilt called My Cancer Genome. And I bring it up here because gene-guided cancer therapy, so targeted cancer therapy, is already a reality. So lung cancer patients, for example, get tested for mutations in specific genes, and based on those mutations, they're often assigned a particular treatment. That's not based on a mutation in like a single gene. So if you have a mutant form of the EGFR gene, your doctor's probably going to put you on erlotinib. If you have a tumor that has this kind of a gene fusion called EML4 fusion, you're going to go on a drug called crizotinib. So that's one of these individualized therapies, right? This is what your tumor says, and you get a drug in response to that. So it seems very clear-cut there. And there's a whole bunch of companies out there that will do this kind of genetic testing on your tumor and give you back a report that will tell your physician if you have a mutation in a gene that has an FDA-ready therapy to go with that mutation. But there's only like a handful of genes. And there's a couple of companies that do this, and we actually just developed our own cancer panel that is now CLIA compliant, meaning we can offer it as a clinical test now, and that's being done out of our Connecticut campus. So a lot of folks are going in this direction now. What's the problem? The problem is that even if you have a mutation in the EGFR gene, you may not be sensitive to erlotinib. So there's this de novo resistance to the drug. So not everybody that carries a mutation is actually going to be responsive to the treatment as expected. We don't know why. And then we've got the problem I just showed you with that 44-year-old patient, where she started to be responsive and then acquired resistance. So it's only getting us part of the way there. It's getting us a good way there, but it's only still part of the way there. The other challenge we have is that cancer genomes are all very unique, right? So if you're one of those patients that doesn't have one of those targeted mutation in one of those genes, where there's a known FDA-compliant target, approved target, and we start looking at your genome and we compare, there's actually very few things that are shared. Cancer genomes are very unique. What do? How do we use that information to decide a potential treatment option? So in addition to building that whole PDX resource I told you about, each one of those tumors gets really deep genomic characterization. We sequence the DNA, we look for mutations, we look for structural variation, we look for amplifications, we look for gene expression. We make millions of measurements on each of these tumors at the DNA level. And We're starting to integrate all of that information, and I can only give you some highlights that I can't go into the details of the actual analysis, but this is one of those plots. It's actually really pleasing. So you can't see, but each of the rows here is a tumor, and they're grouped by their gene expression patterns. And I just want you to notice the blocky patterns here. Right? Can you see the difference between, say, this group of tumors and this one, and this one, and this one? So you can start to see the patterns even if you don't actually know what it all means. And the really wonderful thing for us is that these patterns actually reflect different tumor types. So these are all down here, these are all glioblastomas, these are all brain tumors. They all group together, they all share a common genomic signature. So we can actually pull some really useful information out of data like this. This is just to show you there is coherence at the genome level in these tumors. And we have to come up with a lot of different computational tricks to make sense of these information. And one of the methods we use is this multivariate statistical method called principal component. And really the only thing we're trying to do here is we have hundreds of samples but millions of measurements. And we have to somehow find pattern in that that's meaningful and actionable. So we use statistical methods like this in order to do it. And we compress all of this information into a space where we can look at how the different tumor types group based on their gene expression pattern, for example. And when we do that for these PDX tumors, we find that the groups actually do segregate pretty well, but then we find these unexpected things. So these are all tumors that actually have more mouse in them than human. We can find that just by looking at the genome data. These are all tumors down here that share, they're all different types. So here's, let's see, these are colon and lung and breast. These are all grouping together because they all share this immune signature, right? So these all highly express immune system genes. And we think these are going to be really good models for immunomodulatory therapies. They think we're actually retaining human immune cells in the passage tumors, which would be amazing for doing immunomodulatory therapy experiments. And we've got all this very diverse collection of them that have that signature. So we have to take all this information that we're generating, we have to compress it in a way. And it's been fun to do this because this helps us classify our tumors. But the real question is that meaningful from a targeted therapy point of view? So we've been going back to the literature, and this is a case of a group that used some genomic information to show that triple-negative breast cancer, which is one of these cancers that has no targeted therapy, to actually show that triple-negative breast cancer is not one type of cancer, it's actually at least 6 types of cancers. And if you classify the tumors based on their genome signature, each of those groups has a different sort of therapeutic implication. And this is just starting to emerge, so this is not in standard practice yet. So we have to do a lot of experimentation to see if we can actually translate this basic research into a therapy. And it turns out that our PDX models, we have representations of all 6 of the subtypes, and we're setting them up now on different treatments to see if we can in fact use the genomics, not just to look at the tumors and classify them, but actually come up with treatment plans that are going to be effective for those particular tumors. This is sort of groundbreaking because this is a class of breast cancer that doesn't have a targeted therapy, like I said. And this is just one of the examples of when we compare our PDX models with our genomic analysis with what was published, we get a really high correlation. And we've actually got a number of these mice now up and growing so we can do the gene study. So I want to end with where we are with this concept of avatars. So this really brings everything together that I just talked about. We have the PDX resource, so we have that living bank of tumors, and we've done the deep genomic characterization of all those tumors. We have the tumors in the mice that we've got drug response data for, And we want to bring all of this together to drive individualized prediction, diagnosis, and treatment plans for patients. And so we're doing that in 2 different studies right now, both around this triple-negative breast cancer type. So the PDX for the patient that I showed you earlier, the basic principles of, we're working with Eastern Maine and Maine Med Center right now on that, funded by the Maine Cancer Foundation. And we're doing an avatar for triple-negative breast cancer with Hartford Hospital in North Shore in Evanston, Illinois. So reason we chose triple-negative is that about 15 to 20% of breast cancers fall into this class, and they're called triple-negative because they don't have the receptors that typically fuel a breast cancer, like estrogen and progesterone and those kinds of things. There are no approved targeted therapeutic options, really, for these patients because they don't have these receptors. And while most patients seem to respond well to standard chemotherapy, relapse rates are really high for this cancer type. So with the main study, we're going to create some additional models from patients who have recurrent disease. These are patients where the cancers come back. And then we're going to test the tumor-bearing mice with the same drugs as the patients. So just like I showed you earlier for the lung case and the ovarian case, we're going to do that with these triple-negative breast cancer patients and validate that this platform is going to work for this tumor type. And then we're going to deeply characterize the patient tumor and the PDX tumor genomes, and we're going to correlate the genomics and the drug response to see if we can find additional genomic-based signatures that will predict how different tumors respond to drugs. And then we're actually taking this to the next step in our avatar studies. So the whole purpose behind this, and actually our approval, our IRB approval to do this work, where the information from these mouse-based studies is actually going to go back and potentially inform patient care, our part was approved today, actually this morning, and we're waiting for our partners to have their IRB approved. But the idea is this: these patients are going to come in and they're going to get their diagnostic biopsy, and if they're triple negative, they're going to be consented to go on the study. We're going to get some of that material, and we're going to do our gene characterization, and we're going to create a mouse model for it. Then, in the meantime, those patients are going to go their standard of care therapy. So while the mouse is growing their tumor, they're going to be getting their treatment that they would normally get. And at some point down the road here, the idea is that we're going to have enough time, that the natural history of these cancers provides us enough time that we'll be able to test the second-line therapy options for these patients before their disease recurs. So we'll have that data, unlike that lung cancer patient where we had the data that showed which response would work best for her. We didn't have that in time for her. So the idea here is that we'll get that response data before the patient recurs, so that if she does recur, her oncologist will be able to use that information in part to decide what second-line therapies that she's going to be treated with. So what we're really going to try to do is make this work to benefit cancer treatment for these patients. And we don't know. Will it be useful? The doctor is not going to be required to use the information. They can just use it as one piece of evidence. We don't know if it's going to be useful, but we're about to undertake this and see if we can break through and make it work. And the idea here is, this is the way we're thinking about it currently, where the genomics and the PDX models sort of contribute equally to this whole idea. But really down the road, what we'd love to do is to learn so much about these cancers at a genomic level that we can do this in a predictive, prospective way and not have to rely on these mouse models in the end. So, we have a lot of open questions. Is this going to work? Is it going to be cost effective? Will we know how to use the information to the benefit of the patient? Will it pass all the regulatory standards? Can we make it faster, better, cheaper? These are all the open questions before us. And I just wanted to say that all of this information is available. So all the information we have that's de-identified patient information, we have a database where you can go and you can search for all these models that we have by different criteria. You can get all the information of the type I showed you tonight, including the standard of care, drug response data, all the genomics data are available. So if you're a student and you really want to try something interesting and new, you can get all this data. Maybe you guys can be the ones that come up with the signatures that are going to help us do this work. All this data is out there. It's all public. There's all kinds of analysis tools out there on the web that you can use, and there's nothing to stop you from going out and giving it a shot. So just to sum up, we built this incredible living bank of human cancer models, and that's continuing to grow. And it's a resource not just for us, but we're trying to make this a resource for the entire biomedical research community. That's really the mission of JAX. We're trying to validate this as a platform that can really be used for individualized cancer care. We're going to test that by doing these retrospective studies, by comparing what happens to the mouse to what already we know happened to the patient. Have to validate, have to build this on sound science. But then we're going to actually start to move forward prospectively. Can we use that information in advance to help guide patient care? And then ultimately, can we integrate the whole thing computationally, bioinformatically, to create these predictive models? So we can start with the DNA and get right to the treatment prediction early on without having to go through that long, long time period that we currently are required to do. And then I wanted to end just on one concept that I think is going to be really important for University of Maine Presque Isle and actually all the universities. So there's these things called tumor boards, right? And they happen all the time. They're happening at Cary all the time. The doctors get together, they review what's happening to the patients, they discuss what's going on, they bounce ideas off of each other. We're also doing these tumor boards now where we have basic scientists and genomicists in the same room with the clinicians, because we're talking about discovery, right? We're talking about things where there's no hard and fast answer, where you really have to discuss the pros and cons and what are the weaknesses and strengths of these different methods. So the tumor board concept, we're expanding that to include new models. But I think ultimately what's going to happen in terms of careers is just like you have radiologists, which is a specialty, right? And the radiologist reads all this information from all these amazing imaging technologies, and they actually have the background to interpret them, and they communicate and consult with the physicians who may not be experts in this radiology. We think that basically there's going to be the radiologists of the genome. That there's going to be positions in hospitals of people who not reading X-rays, but who are reading all of this complex DNA information that we're generating, who are going to help sort of interpret it, package it, and work with clinicians in a way that we can actually deliver on the promise of individualized medicine based on genomes and PDX models, and do this in a robust, scientifically valid way. And so this is going to be like a whole new industry, a whole new field that's going to be opening up, I think, in the next decade or so. And we have to make sure we're educating our students to be ready for this future, because I don't think we necessarily are right now. And it's amazing how rapidly these changes are happening. The landscape in this field is changing almost daily. So with that, I want to thank, there's so many people to thank. Thank you for spending the evening to hear about this work. I obviously don't do this by myself. This is a huge team effort. Many, many different disciplines coming to the table. The patients who actually sign the consent forms to make their material available to do this kind of research, we could not do it without them. We could not do it without our clinical partners. We could not do it without the support currently of a lot of foundations in Maine and elsewhere. And with that, I'm happy to take any questions you might have.
Yeah, you mentioned, since my students have been studying buffers, you mentioned when a piece of the tumor comes out of the patient, before it goes into the P0 mouse, it's put in a buffer solution. Did you happen to know what the composition of that buffer was?
It's called RPMI.
RPMI.
It's just a standard kind of saline buffer.
Okay.
And so what happens is when the surgical material comes out, and it first has to go to the pathologist because the patient's care goes first. The
Yeah.
pathologist takes all the material they need to make the diagnosis. And then we get what's left over, and they put that in that buffer on blue ice. They put it in a FedEx box, and it goes to California, and it goes in a mouse. Immediately. We have people we have people on standby like 24 hours a day to receive the material. So, yeah, Steve.
How do you keep your NSG mice free of infection?
Yeah, so that's called health status. So these mice are in specially designed cages, in specially designed rooms. And the people that go in and out of these rooms, I've done it, I wouldn't want to do it every day. You basically have to take off all your street clothes. You go through what's called an air shower. So, I don't know, was it a Andromeda strain where they had those? So you basically have to go in this room and you go like this, and there's all these air showers coming down on you, which takes off all your dander and all that stuff. And then you put on gown, a fresh gown. You put on sterilized shoes, you put on a mask, you put on a bonnet, you put on goggles. It's not quite like the moon suit with a respirator behind, but it's pretty darn close. So this is not easy or fun work to do, I'll tell you that. The technicians who spend all their day receiving this material and putting it in these mice so we can do these studies, they are amazing dedication.
And so far, you've had no disasters with something wiping out a population.
I wouldn't say that. I mean, every once in a while, was it in Jurassic Park, biology finds a way, you know, there's movie references. So, with no real disasters, but there certainly have been cases where mice have gotten sick from something that, you didn't expect them to get sick from. So it happens, biology finds a way. So, yes?
As far as the drug testing on the mice, do you use just FDA-approved drugs, or do you also stay in touch with research companies, biology companies that are doing research testing on
That's
patients and so on?
really great question. So, when we're doing the standard of care, so when we're trying to match what we do in the mouse to what the patient got, that's usually all federally approved standard of care drugs. But for example, we've been working on it for a year, we just signed an agreement with a pharmaceutical company who wants to overcome, new drug combinations to overcome EGFR resistant, erlotinib resistant EGFR mutant lung cancer. So this is that 44-year-old patient I showed. This is all about her, right? Her tumor became resistant. We want to know how to overcome that resistance. They think they have a drug combination that's going to overcome that resistance. We're testing that in these mice. We're actually setting up the mice now. It's going to be another couple months before we can do the study because it takes months for the tumors to come online. So we are working on new drug combinations and new drug types with pharma in a research context, so we'll be able to publish this. This is not just a for-profit contract type of project. This is actual research. So we'll be able to make it public when it's all done. It'll be really fun.
And like you say, the timing of it seems to be a lot quicker using the mice versus, a study using actual patients.
Definitely, and cheaper.
Yeah.
It's all expensive, but it's cheaper than doing this in clinical trials on people. Yeah.
What platform are you using for the genomic characterization of the tumor?
Several different ones. So Illumina, it's for the DNA sequencing and the RNA expression, gene expression, it's all Illumina based. For our panel, our cancer panel that we just finished designing and got CLIA compliance for, that's adjuvant exome capture based stuff. And we still use some arrays to look for copy number variations. So sometimes in cancers, certain genes, you know, the genome goes kind of crazy, and certain genes get what we call amplified, so they get lots and lots of different copy numbers, and there's too many of them, and then the cells become hyper-responsive to signals. So these are all signals these cells normally see in some context. It's just that they get hyper-responsive to them. So we look for that kind of stuff with standard array technology. It's changing all the time. Pretty soon, it's going to be cheap enough, we're just going to sequence the genome. Right? That's coming. We don't have the analytics worked out for that yet. So even though genome sequencing is becoming cheaper, we don't know what to do with the data yet. But we're working on that in the background so that we're going to replace what we're doing now with whole genome tumor sequencing probably in the next 2-3 years. Yes, sir.
Yeah, I have a two-part question. Would this be considered gene therapy?
No.
How are healthcare companies willing to pay for this treatment?
So that was, I think, one of my questions. So, yeah, oh man, you know, I'm a scientist, and it hurts my brain to think about insurance companies and how how that hurdle's going to be overcome. So the gene testing, that single gene panel testing, a lot of that now can be covered in specific cases, and the oncologists in the room can probably speak to that more than I can. But getting this kind of mouse-based stuff done, this is all still research, all clinical trials, all funded either by federal government grants or foundation money. And we don't know how easy that work is going to be able to be reimbursed, for example, by insurance companies. It's going to be an uphill road for sure. But I think we can still make tons of insights that are going to impact patient health before that happens.
I just know that my healthcare plan has a specific exclusion for gene therapy.
So yeah, so gene therapy, it depends on, when I hear of gene therapy, I actually think of trying to replace a broken gene with a fixed version of it. That's what I think of when I think of gene therapy, so, which is very different than the kinds of things we're doing here. We're characterizing the genome, but we aren't replacing the genome with anything, so we aren't actually doing gene therapy. It's really using genomics to drive drug therapy, in this case. Yes, Sean?
Of the organizations that are working on this, how collaborative is this, or is it very silent?
That's a good question. So there are, there's actually a fair amount of collaboration. This type of work, there's not a lot of competition to do it in academic because the infrastructure you need to house the mice, to train the people to do this kind of work, and then to have the computational biologists who can do the analysis to come up with the genomic-based signatures that you use to, all of that stuff is highly specialized, and it's very rare to have it in an organization like the Jackson Lab. So we get a lot of collaborations because a lot of people can't set up the infrastructure, but they want to work with us. So we get a ton of really great collaborations just because of the unique nature of what we have here in Maine. That we can extend out to the rest of the world. There are other groups who work on patient-derived xenografts. They build small collections of very focused tumors. No one really does the scale and scope and diversity of what we're trying to do, but there are smaller pockets of them out there. And there's one commercial group in New Jersey called Champions. And Champions is doing a PDX patient where they put your tumor in the mouse and then they test the drugs and they give that result back to your oncologist. They are a commercial entity. They don't make any of their data public. So they're doing this, but it's all done in a proprietary fashion. They don't share, and they're not using NSG mice. They're using athymic loop mice.
For future avatars, are you using only academic centers to collaborate with you, or are you out in the community with some of the cancer centers
we partner with a lot of, right now it's like 25 different organizations, and we're actually about to, for the breast cancer work I talked about, we also have a colon project that we're just about to start with the Southwest Oncology Group that will mean bringing in additional partners. So as long as, so our one criteria for this work is that we will partner with anybody as long as we can make the models available to the scientific community. Right? This is our mission. Our mission is not to build a proprietary collection of things that we profit from. It's really to build this resource to drive what happens in the biomedical research community, share as broadly as possible. So that's the only restriction that we have. You want to work with us, that's wonderful, but nothing proprietary, because that doesn't really fit with our mission.
Does that, how does that work in regards to the various drug companies that you work with?
So that is, so we don't give, so what happens in that, so this is all the kind of contract research stuff that goes out at Jack's West that's not part of the PDX program. That is an entirely separate part of the organization that intersects with pharmaceutical companies. So that is a part of what I talked about. Anything that's part of what I talked about gets made publicly available. There are studies that are done by us for a company. It's usually testing a specific new drug on these mice. The company pays for the research and they get the data and then they can decide what to do with it. But if they want to really partner with us and cost share and do all this stuff, then just the drug company I just talked about tonight, this is a collaboration. They're going to make the models available. They're going to make the data available. That's the model we prefer to work on for this particular program. So I don't have anything to do with the contract part of what Jack's West does in Sacramento at all. It's not anything I personally want to do. It's not that I'm against it. It's a very necessary part of the industry. No problem. I just don't want to mix that model with what I'm trying to do. Yeah, Jean.
Is there anything you can learn, not from the tumor, but from the patient's DNA, just straight where they have the tumor? Or can you learn something about how they might respond?
From germline DNA?
Yeah, or just regular.
So, does anybody know why we wouldn't want to sequence patients' germline DNA and be responsible for it? Any ideas? Because it's identifiable, right? So, if we go into the area of we decide to sequence all you people in the front row here, that brings up all kinds of patient protection issues, right? About patient privacy, and you have to deal with the data in a very special way, right? To protect them, and it just opens up a whole sets of regulatory issues. And we really don't know how to interpret the data anyway, right? So kind of what we're talking about today is if I sequenced everybody in this room, we would come up with lots of variation in the DNA, and we don't know biologically what it means. I wouldn't know what to tell you about it. I could describe it to you, but I wouldn't know how to help you understand that so that you could make informed health decisions. We just aren't there yet. But then I'd have all this identifiable patient data that I'd have to be very protective of and conform to a lot of federal regulations to make sure we didn't release it. Tumors aren't you, right? These tumors are things that have acquired mutations that are different from your germline. So we don't have to worry that somebody's going to look at a tumor DNA and be able to pin it back to an individual. So we don't have those same kind of worries about making those data publicly available. Yes?
So that private company in New Jersey, Champions, if I had the money and I had cancer, I could pay them to give my cancer to a bunch of mice and tell me what cured those mice so that I could drive my own care? Is that something?
Your
you
doctor
actually
would have to do that.
My
So
doctor,
they,
right.
yeah, they wouldn't do that for, I doubt they would do it for an individual, but your oncologist could work with them to build a model, a mouse model for your tumor. And I don't know what criteria they use, and I don't know if they restrict the different tumor types that they actually accept in that program. But yeah, you would probably have to pay out of pocket
Right.
a couple $1,000 they subsidize it, so it costs us about $60,000 to $80,000 to create one of these models and do everything I just talked about. It's not inexpensive. That's right now. So who was I talking to today about the cost? So the way I see the cost is that, remember how much the federal government invested in developing GPS technology? So the first GPS probably cost a couple of $2 billion. And now you can buy one on Amazon.com for $80. And that's just because it takes investment in a technology to run it through its paces. And then once we know more about how to work it, then we can, faster, better, cheaper. That's what we're working towards. But we have to do this upfront work. It's like building the GPS. It's going to cost a lot. We know that. But the insights we get will eventually lead to products and ideas that we think will make this really much less expensive, much faster, and easier to deploy to many, many different types of patients.
So do you envision a day when that is a regular thing, not someone paying out of pocket, but any person with any insurance or lack thereof, their oncologist will be able to take a piece of the tumor and say, well, we're going to send this off, and within a short amount of time, the mice will tell us what we should be doing for you?
Yep. If you had asked me like 10 years ago, did I think we'd have single gene tests that would be used to drive patient care like we have today? Well, maybe 10 years ago I would have, but this is happening very, very quickly. So yeah, I think we'll get there, but I don't think we'll get to that point for quite some time. There's a lot of research that has to be done yet, but sure, that's the vision. That would be outstanding. And even better would be to use the mice to do something I haven't talked about that my research group works on, which is early detection and also looking for susceptibility up front so that you get all of your screenings and stuff earlier and earlier if you know that you're part of the susceptible class of people. So, there's all kinds of things I think we can do other than intervene at this late stage in cancer development. We've got to get better at prevention and early diagnosis. And so there's all kinds of research happening on that as well, but I didn't talk about that tonight.
Do you envision a point where there's a vaccine available, much like polio and so on?
And so we talked about that a little bit today with the students as well. And I don't know about vaccine, but I can tell you that there's been some tremendous strides in immune-based cancer therapy. It was like the discovery of the year last year in Science magazine, which is our big, big high-profile magazine. There are going to be, I think that harnessing one's immune system to target these cancer cells is definitely going to be one of the things we want to do, which is why we were so excited to see that we have models that seem to have preserved human immune cells. Because how are you going to study how to make this possible if you don't have a model system? Well, it looks like we've got a chance at having this really great set of tumors that have this property that we can start to explore how best to harness somebody's immune system to fight the cancer. That's pretty amazing. So we don't know yet what that's going to lead to, but we're working right now on validating that the cells that we're seeing in these mouse, in these engrafted tumors are in fact human, and that they're what we think they are. And if they are, we're going to be doing some interesting immune-based therapies on those models. But I don't know if it's going to be a vaccine. I really don't know how to think about that right now. I'm not an immunologist. It's stuff goes on. Great. Hey, thank you all again for a good I went to the science and mathematics school, I went to Cary, I saw so many different people today. It was just a real pleasure, and I so appreciate everyone taking the time, and so thank you. I hope to see you again.
The skis?